AAV5 capsid, lung specificity. The gene delivery system to be used will be adeno-associated virus helper-free system. For liver cancer, in the triple transfection for RAAV production, we use a Rep2/Cap3 plasmid supplied in trans. This plasmid has genetic elements of the AAV2 genome, chromosome 19 site-specific integration, and AAV3 capsid, liver specificity. The gene delivery system to be used will be an adeno-associated virus helper-free system. After the human development phase, most cells in the human body reach a state of quiescence, GO phase, referred to as the G0 phase or resting phase, and do not divide further by exiting the cell cycle. Quiescent cells can re-enter the cell cycle. The development of tumorigenic cancerous cells occurs when they bypass quiescence and proliferate. In the typical cell cycle of G1-R-late G1-S-G2-M, there is indication that loss of regulation in R checkpoint would allow cells to enter the cycle in the absence of mitogenic signaling, leading to unscheduled proliferation. While it may be unfeasible to completely convert cancerous cells to normal cells, we would want to arrest the proliferation of cancerous cells by introducing a therapeutic gene of interest, GOI, into them that will convert them into quiescent cells. In the G0 phase, it has been found that the protein, which is a member of the retinoblastoma family, viz. P130, binds to the DP-E2F protein transcription factor and inactivates it. The P130/RBL2 retinoblastoma-like protein 2 is a 130 kilodalton protein. The P130 gene is a potent inhibitor of E2F-mediated transcription activation and may act as a tumor suppressor. In a study, the gene was inserted into retrovirus vehicle and introduced into the human lung cancer cell line in mice. It was found to suppress tumor growth. The P130 gene has been obviously isolated and its genomic structure has been determined. P130 is a bona fide tumor suppressor gene and it may be a candidate for cancer gene therapy for lung cancer and liver cancer. Conclusion: the P130 tumor suppressor gene will be the therapeutic gene of interest in our study, and we would like to insert it into the RAAV vector. After completion of triple transfection, RAAV variant particles containing a P130 gene in the DNA and a Rep2/Cap5 lung specificity or Rep2/Cap3 liver specificity capsid are created. Thereon we will inject into the bloodstream, intravenous or intraarterial or intramuscular, this recombinant adeno-associated virus into the central nervous system for cancer cell line patient, lung cancer and liver cancer, and wait for integration of the virus carrying the therapeutic gene of interest onto the specific site on human chromosome 19. The P130 gene will be expressed from human chromosome 19, and the P130 protein will bind to and inactivate the DP-E2F protein transcription factor and basically arrest the proliferation of the cancerous cells by converting them into quiescent cells. Cancer, apoptosis, programmed cell death. High energy rays, radiation, damage DNA in cells, causing them to die or stop dividing. Since cancer cells divide more frequently than normal cells, they are more susceptible to damage. Healthy cells can be affected as well, but are better able to repair the damage. 3H hydrogen radioactive thymidine labeling of DNA triggers apoptosis potentiated by labeling of the plasmids. Potentiated by E1A adenoviral protein. E1A adenoviral protein is a 32 kilodalton protein. I propose